Hey there, YouTube, people, universe, all the things. You guys know what you're called. It's Thursday morning, because I could not get the video done last night, but I did get it done, or at least started, we'll see. So, research vlog number 1. Pretty much just gonna talk about what I do, because I don't think I've quite outlined the project yet. Seemed like a good place to start. And probably in one take, because I don't have time to edit this and I kinda need to get to class. So, what do I do? I study squid, we've covered that at least, and how they're affected by ocean acidification. So I'm looking at the physiological and developmental effects of ocean acidification on the early life stages of a local species of squid near Cape Cod, the Atlantic longfin squid, Doryteuthis pali, Latin names. Really, I look more at the paralarval stage, which is the first hatchling stage of squid. Why look at the early stages as opposed to the adults? Well, early life stages of animals are typically a lot more sensitive to environmental change than the adults are. Once you're an adult and everything's kind of established, you're a lot more hardy, you have a lot more energy stores, you're just able to better handle or deal with stress. Rather than, you know, when you're a baby and if the world crushes you, you pretty much don't have a choice in the matter. We get the squid and then take them to the lab, they lay the eggs in these nice little mats. So each little egg capsule contains like 50 to 200 eggs. And I take a couple of those capsules and put them in a cup that has a specific CO2 level in the water so that they're exposed to an experimental ocean acidification condition. To explain ocean acidification, if you're not really familiar with it, it's a large part of the climate change problem as we're pumping more and more CO2 into the atmosphere because of fossil fuels. More and more of it is going into the ocean, and when CO2 enters the ocean, it goes through this buffering reaction where it transforms into carbonic acid, but more often not more often, its more stable situation is where it's in a buffering situation as bicarbonate. So that means that the carbonic acid has let off a hydrogen ion, and the bicarbonate is there to act as a buffer. But it means that overall there's an increase in the amount of hydrogen ions in the ocean, thus it's getting more acidic, and thus ocean acidification. So what is it that I do? Once I've put the eggs in the OA conditions, I raise them till hatching, and then I take the paralarvae that hatch like immediately and examine them for physiological changes, for developmental changes, so morphology, the size and shape of the body, the amount of yolk sac that they're born with, the amount of time it took to hatch. I also look at embryonic survival. I'm going to be looking at respirometry, so the heart rate, well, not the heart rate, but the oxygen consumption rate, so the breathing rate of the squid in the OA conditions. We're going to be looking at gene expression, so is there a change in what proteins they're producing in these environments. We look at the statoliths, which get dissected out, and then we're looking at structure as well as size, crystal structure, chemical composition, density, so a lot of things with that. We're going to be looking at swimming behavior and phototactic response specifically as part of swimming behavior, because the statoliths are very important in their ability to orient and direct, so if the statoliths are being degraded, there is the potential effect that their swimming is also degraded. Last year I did a dose-response curve that I'm finishing up this year, looking at these physiological effects over a range of CO2 levels, and then this year I'm finishing that up and then also moving on to do more set levels that correspond with these respirometry and behavior trials, so that it's a very consistent range of CO2 levels that we look and repeat over the season to also get like a seasonality picture. Also going to do a lot of stuff in situ where I look at the chemical oceanography at the squid breeding sites, so I'm going to be looking at the pH and CO2 levels at the places where the squid are gathering and laying the eggs to see like what exposures the natural system is presenting, because it's already a point to which it turns out in the coastal system pH can really spike down and the CO2 levels can spike up to really, really extreme levels that we haven't predicted for the open ocean for like 100 years. So that's why it's relevant. That's what I'm doing. Yay, science in one take. Woo! Catch you guys later. Lots of things. Lots of things. Ooh. Vitamin C allows me to talk quickly in the morning. Ooh, could not live without it. Well, I could, but who would be want to? There will be like subsequent videos that more thoroughly and are scripted explain all of these portions and concepts in the future.